Good day, this is the Professor, drwillphd, PhD, and today is Thursday, April 27, 2023. It is 10:08 and 24 seconds, p.m. Eastern Daylight Time, and today's research, cancer diagnosis. So let's jump to it here. A simple test can offer early cancer diagnosis. The new diagnostic, which is based on analysis of urine samples, can also be designed to reveal whether a tumor has metastasized. Date April 25, 2023. Source: Massachusetts Institute of Technology. And here's designed a nanoparticle sensor that could enable early diagnosis of cancer with a simple urine test. The sensors, which can detect many cancerous proteins, can also be used to distinguish the type of a tumor or how it is responding to treatment. The nanoparticles are designed so that when they encounter a tumor, they shed short sequences of DNA that are excreted in the urine. Analyzing DNA barcodes can reveal distinguishing features of a particular patient's tumor. The researchers designed their test so that it can be performed using a strip of paper, similar to an at-home COVID test, which they hope can make it affordable and accessible to as many patients as possible. They are trying to innovate in the context of making technology available to low and middle resource settings. Putting this diagnostic on paper is part of their goal in democratizing diagnostics and creating inexpensive technologies that can give you a fast answer at the point of care. In tests in mice, the researchers showed that they can use the sensors to detect the activity of 5 different enzymes that are expressed in tumors. They also showed that their approach can be scaled up to distinguish at least 46 different DNA barcodes in a single sample, using a microfluidic device to analyze the samples. As for several years, the laboratory has been developing synthetic biomarkers that could be used to diagnose cancer. This work builds on the concept of detecting cancer biomarkers, such as proteins or circulating tumor cells, in a patient's blood sample. And these naturally occurring biomarkers are so rare that it is nearly impossible to find them, especially at an early stage. But synthetic biomarkers can be used to amplify smaller scale changes that occur within small tumors. In previous work, researchers created nanoparticles that can detect the activity of enzymes called proteases, which help cancer cells to escape their original locations or settle into new ones by cutting through proteins of the extracellular matrix. The nanoparticles are coated with peptides that are cleaved by different proteases, and once these peptides are released into the bloodstream, they can then be concentrated and more easily detected in a urine sample. The original peptide biomarkers were designed to be detected based on small engineered variations in their mass, using a mass spectrometer. And this kind of equipment might not be available in low-resource settings, so the researchers set out to develop sensors that can be analyzed more easily and affordably using DNA barcodes that can be read using CRISPR technology. And for this approach to work, the researchers had to use a chemical modification called phosphorothioate to protect the circulating DNA reporter barcodes from being broken down in the blood. And this modification has already been used to improve the stability of modern RNA vaccines, allowing them to survive longer in the body. And similar to the peptide reporters, each DNA barcode is attached to a nanoparticle by a linker that can be cleaved by specific proteins. If that protein is present, the DNA molecule is released and free to circulate, eventually ending up in the urine. As for this study, the researchers used 2 different types of nanoparticles, one a particle made from polymers that have been FDA approved for use in humans, and the other a nanobody, an antibody fragment that can be designed to accumulate at a tumor site. Once the sensors are secreted in the urine, the sample can be analyzed using a paper strip that recognizes a reporter that is activated by a CRISPR enzyme called Cas12a. When a particular DNA barcode is present in the sample, Cas12a amplifies the signal so that it can be seen as a dark strip on a paper test. The particles can be designed to carry many different DNA barcodes, each of which detects a different type of protease activity, which allows for multiplex sensing. Using a larger number of sensors provides a boost in both sensitivity and specificity, allowing the test to more easily distinguish between tumor types. In tested mice, the researchers showed that a panel of 5 DNA barcodes could accurately distinguish tumors that first arose in the lungs from tumors formed by colorectal cancer cells that had metastasized to the lungs. The goal here is to build up disease signatures and to see whether they can use these barcode panels not only read out a disease but also to classify a disease or distinguish different cancer types. As for use in humans, the researchers expect that they need to use more than 5 barcodes because there is so much variety between patients' tumors. And this kind of testing can be used not only for detecting cancer, but also for measuring how well a patient's tumor responds to treatment and whether it has recurred after treatment. The researchers are now working on further developing the particles with the goal of testing them in humans. They performed phase 1 clinical trials of an earlier version of the urinary diagnostic particles and found them to be safe in patients. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.